Structure of seminiferous tubule. At the periphery, towards the basement membrane, spermatogonium. Spermatogonium divides to form primary spermatocyte followed by secondary spermatocyte. Secondary spermatocyte changes into a spermatid. Towards the lumen, spermatids develop into spermatozoa. Supporting cells, sustentacular cells or Sertoli cells are also seen. They are tall, columnar cells with large nuclei. They have many interdigitating processes to support the germ cells. They form blood-testis barrier. They also produce inhibin hormone and Mullerian inhibiting substance. Interstitial cells of Leydig are also seen. These cells produce testosterone. These are polygonal or pink cells. Sertoli cells create junctional complexes to form blood-testis barrier. The interdigitating process of Sertoli cells form tight cell junctions separating the germ cells from each other. However, they do not allow the blood cells from the periphery to move towards the luminal side. The process of spermatogenesis begins after puberty. Hence, spermatids are like foreign bodies and blood cells do not identify them as a component of body. Therefore, it is important to have blood testis barrier so that no autosomal autoantibodies are formed against the sperms. Inhibin hormone is produced by Sertoli cells. Inhibin gives a negative feedback to the pituitary so that pituitary reduces the production of FSH. During the development, Sertoli cells produce Mullerian inhibiting substance which suppresses the Mullerian duct or female duct. Hence, the Wolffian duct and male sexual characteristics develop. Female genital system. Ovary. Ovary releases the secondary oocyte, which is taken up by the fallopian tube. Fertilization occurs in the ampulla of fallopian tube. Zygote moves through the fallopian tube into the uterus, where implantation occurs. Lining of the ovary. Cuboidal epithelium. Peritoneal covering. Primary oocyte is located in the periphery of the ovary or cortex of the ovary. Neurovascular bundle or ovarian blood vessel and nerves is coming into the medulla. Follicles are seen in the cortex of the ovary. Types of follicles: primordial follicle, primary follicle, secondary follicle, tertiary or Graafian follicle. All these have primary oocyte. Primordial follicle. It has primary oocyte covered by simple squamous cells. Primary follicle. The primary oocyte is covered by a single or multiple layers of simple cuboidal cells. The simple cuboidal cells are also known as granulosa cells. These granulosa cells contributing to forming zona pellucida around the oocyte. Granulosa cells also produce estrogen with the help of theca cells. Theca cells lie outside the basement membrane of granulosa cells. Secondary follicle. It is also known as antral follicle. The primary oocyte is covered by granulosa cells and the follicles also developed many small antral spaces. Graafian follicle. The primary oocyte is covered by zona pellucida and granulosa cells, which are now called corona radiata. The antral spaces combine to form one large antral space. Granulosa cells covering the corona radiata and form the connecting stalk cumulus euphorus. Just before the ovulation, primary oocyte changes to secondary oocyte. On ovulation, secondary oocyte is released from surface of ovary. The secondary oocyte is covered by zona pellucida and corona radiata. Cumulus euphorus cells are left behind in the ovary. The remnant of Graafian follicle changes into corpus luteum in second half of menstrual cycle. Corpus luteum produces progesterone hormone. In case secondary oocyte is not fertilized, corpus luteum regresses.